So our next speaker is Nir Bental. Nir had gotten his bachelor's in physics and chemistry from Hebrew U, Nir?
Yes, and also
Yes.
biology.
Right. So, and then his PhD in chemistry from Technion, and then he did his postdoc at Columbia. In 97, he had accepted a position at Tel Aviv University. And so that would have been, so he would have been at Columbia before I was there. And the other day I was trying to figure out when I met him for the first time, and I couldn't. It must have been in the early 2000s. But the reason I couldn't is because his work and Nir had been present in my scientific life for such a long time that I can't figure it out anymore. Nir works in the structural biology space. He builds tools such as Conserve, so many of you may have used it, but also he's just exploring the structural space over there. His research interests range from anthrax to autism to drug development and just generic exploration of the protein space. He's been recognized by many agencies. The latest prize that I saw on there was a NATO 60th anniversary Science for Peace and Security Prize for his work in anthrax neutralization, I think, right? And it's really super interesting stuff. But today, to you, he is going to talk about protein archaeology, which I can't wait to hear about, and about basically copy pasting pieces of proteins to make what we have right now. So Nir, please.
Thank you very much for the invitation, and I'm happy to talk. So as you can see, it's joint work with a lot of people, including Rachel Kolodny from University of Haifa. And the idea is to try to look at protein space and figure out what can be learned. And it's not important to mention here, I mean, because everybody knows that protein play a central role in living organisms, and their emergence and the emergence of life itself are interconnected. But I'll just briefly mention it nevertheless. So, and there are various chicken and egg conundria. So, for example, proteins are coded by DNA, and DNA itself is synthesized by proteins. So, what came first? And perhaps it was RNA. But to study proteins, to study how proteins emerge, people like Salma, as we heard, they do prebiotic chemistry. So starting from Miller kind of experiment with heating and electric shock and heating and wetting and cooling, where from simple atoms you find small compounds and eventually amino acids that maybe polymerize into peptides, and hopefully proteins emerge from all that. And I'm taking an archaeology-like approach using current protein repertoire to try to figure out what happened before. And that's more similar to the approach that Vic presented and also a part of Yana's talk. So that the idea is that at some point in time, there were a few sequences maybe of proteins, and because of various evolutionary processes, they multiplied and over time, there are more and more sequences. Now, of course, most of these are not even stable, and they didn't survive because of structure issues and other biophysical issues. And then many others maybe were stable, but didn't have function, so there was no reason to keep them alive, and only some survive all these, both filters. And then we are left with proteins today, our current protein repertoire, and again, a lot of them, they are unstable or dysfunctional, and they are therefore discarded. And then we also, for some of them, we know the structure. So these are marked with squares. And for some of them, we know function to some degree at least. And for some of them, we know both. But then we don't know all the protein that emerged over time. Those were discarded. We don't know them anymore. We just have what we have today. And based on this repertoire, which we call the protein universe, we can address two different questions, different but related. So the first is how proteins evolve. And then the more difficult questions is how did they emerge? And it's more difficult because it means looking far back into evolution, and the signal, a lot of the signal may have been discarded, so it's difficult. Now, how do we represent a protein universe? And usually people start with proteins of known structure, so I'll be following this tradition also here. And there are three different issues here that are related to each other. The first of them is what kind of entities? So are we dealing with full chains or supradomains or domains? And the other one is the similarity measure. Will it be sequence or structure or maybe both? And whether we will be asking for similarity across the entire thing or just locally. And then the kind of representation. So a common choice is domains, because we know that they are repetitive entities. So why bother with full chains when domains are already defined? Although I will question this paradigm, and you'll see it in a while. And then the other issue is what kind of similarity measure, as we said. And for example, to make sure that we are dealing with divergent rather than convergent evolution, maybe sequence similarity is beneficial. Or if we care to look at the convergent evolution, or also to a longer evolutionary distance where the signal may have been discarded, then maybe we should look at structure similarity. And maybe we want to look at both to be on the safe side. And then there is the issue of representation, where the most common type of representation are lists, which is the kind of representation used in the domains databases like CAS, SCOPE, or the more recent ECODE. But there are other ways, and it's very, the domain databases are very useful when you care about a specific protein family and you want to look at your nearest neighbors. But then if you want to look at for relationship between different families, the list representation is not particularly informative. And then people try other means. For example, points in 3D or 2D space, where each point would be a domain, for example, and then the distance would correspond to their similarity. And then you can color code it by some qualities. Here it's by secondary structure. And we will be using graphs, and I'll elaborate on that. But just to make sure that we didn't invent all of this, but we just contributed. So here are references. So you can see on the left, references for representations using point in some space, 2D or 3D. And then on the right are some references for networks. And of course, I'm missing a lot of other work, including some of the works in previous talks. So I'll be presenting a network view of the protein universe. And I'm going to cheat a little in that over time, as databases improved, we changed the database, but the conclusions that I'll be presenting, they are not very sensitive to the kind of database. So just to be concrete, I'm going to present the first data set that we used, and let's pretend that the rest are the same, although they are usually larger. So we started, and that was maybe 10 years, even more ago, with a set of nearly 10,000 scope domains, reduced to 70% sequence identity to eliminate unnecessary redundance. And then we compared them all against all over sequence, based on sequence similarity. And we asked for similarity over long enough residues so that we believe that the signal is real rather than just being random noise. And we used a graph representation. So we see nodes that are scope domains colored by scope class, and edges connect sufficiently similar domains, and we present the result using Cytoscape and our own Cytoscape app called Cytostruct that allows visualizing the structure and easily combining Cytoscape to your visualizer, for example, Pymo. So this is a concrete example, just a very small, like a tiny network that shows 4 domains, and you see so each of those is a domain, and you see similar parts that are marked with similar colors. So the similarity between these 2 domains, it doesn't encapsulate the entire domain, it's just a blue part, and between those, there is this orange part, and the link between them gives the values. So in this case, it's 86 amino acids with 30% sequence similarity and below 2 angstrom RMS deviation. And here it's also similar and so forth, but this is an arbitrary choice. So, and with that, we looked at all the domains in this set, and the result, of course, depends on the threshold that we choose for similarity. So when we use very lax threshold, then essentially everything is similar to everything. And in this case, it's more than 55 amino acids with more than 30% sequence similarity. But in the other extreme, if we use very stringent threshold in green, and here we ask for more than 75 amino acids with more than 50% sequence similarity, we see that essentially there is no network, just isolated islands. Now, it's actually interesting to look at all of them, depending on the threshold. And so here is a very lax threshold. And as we use more stringent threshold, we see that there is a decomposition into 2 regions, like a large connected component and the smaller islands. And again, and as we use more stringent threshold, it means that we are looking, that we are evolving. We look closer and closer in evolution to current day. And this is like the most recent evolutionary events, if you want. But, and it's actually interesting to follow this process. It can be informative, but we want to look at one typical case just to see what protein space look like. And it's an arbitrary choice, but I stop here. And for what I say, even though this is an arbitrary choice, the conclusion doesn't depend very much on the threshold. And you can see here the kind of protein space. It's color coded based on the type of secondary structure. So blue is proteins of the all alpha type based on scope. Red is all beta and so forth. Green is alpha/beta. And the interesting observation here is that there is a large connected component living together with isolated islands here. And I'm not showing it, but each of those isolated islands actually correspond to a protein family. So maybe hemoglobins or so. And this brings in an old question. So people from the beginning of domain evolution, if you want, or the emergence of when people notice the presence of domains, of those repeating elements, people thought of them as individual and disconnected families. But over time, people saw that there is so-called cross-fault similarity between domains of seemingly different origins. And over time, as people saw more and more of those, people were debating on whether protein space is continuous or discrete. Now, mathematically, this is not well-defined concept, but the network view gives a straightforward definition of this. We can think of the large connected component as continuous space, because here all the domains are evolutionarily linked to each other. So that is not the entire domain are similar, but part of them are, and these would be the cross-fault similarities. So that continues. And then there is the isolated island that correspond to the discrete region in protein space. And interestingly, they live side by side in contemporary protein space. So protein space is both continuous and discrete. Now, of course, from evolutionary standpoint, and you can see that the large connected component, the continuous part of protein space, has a lot of alpha/beta proteins like Rosemond and so forth, which are known to be ancient. So that's an interesting part of protein space from evolutionary standpoint. Now, this large connected component can be viewed as a collection of evolutionary pathways linking various groups, and I picked just one of them. You can see it connects 8 different domains from 6 different folds, including team barrel and FAD, NAD binding domains, and so forth, that are in flavodoxin, that are all known, and Rosemond, and all of them are known to be ancient domains. So, and they are right in the center of this continuous region in protein space. Now, I don't know how it goes. So I don't know, it could start in this protein and going from all the way to the other end, or the other way around, or it could start somewhere in the middle and propagate in both directions. That information I don't have. I mean, this analysis doesn't readily yield it, but it gives indications that like hints to start looking for. Now, the next thing that we tried to do was to figure out what is it that is responsible for the similarities in protein space. So we looked for the shared segments that are responsible for the similarities, and we call these themes because other names were already taken, including domains and motifs. And we figured out a way to look for them. We just looked at the alignment and then looked for the shared segment. There's a dynamic programming way to do it, but I'm not going to present it because it was published already. But the important thing is that we can count them. So, for example, themes of 35 amino acid long, we can see that here is a set of themes with variations, 7 of those within our set, and here is a theme with 5 variations and so forth. This is for 70 residues, and again, we can count them, and then we can look at them, and here is an analysis of this plot. So this is the number of themes as a function of the size of the SYM cluster, and this is for domains, and this is for whole proteins. So, and you can see short themes of 35 amino acids, are a lot of them, with two appearances, and very few of them with many appearances, up to several hundreds. So this is like a power law or semi-power law distribution, which is typical for essentially anything in nature. And you can see that reuse increases as we look for smaller things. I want to spend some time about the analysis for whole proteins, and here again, large reuse with of small themes and less reuse with larger theme. And in particular, let's look at themes of 100 amino acids. Now, domains are typically 100 amino acid long, and this is the most common form of reuse. So we were expecting something special happening for reuse of domains, of segments of 100 amino acids, but we see that it's not the case. We see a little bit more reuse of those segments compared to segments of 125 amino acids, and less reuse compared to segments of 85. But it's not. There is nothing specific there. So domain, in our view, it's just one type of theme that people noticed, but it's just one of many forms of reuse. Another quality of our themes is that, unlike domains, where they were reconstructed under the assumption that any amino acid can belong to one particular domain, not more, but we didn't ask for that in the case of themes. So an amino acid can belong to as many as possible themes depending on the degree of reuse. And that leads to very complex pattern of reuse, shown here with a Russian bell pattern. For example, that is observed here with the propeller structure here. This is a 7-blade propeller. And if we look here in pink, we see that this single blade marked in pink, it is shared by many domains, all the domains with the purple frame. So maybe more than 30 or maybe 40 domains in our set. And then if we look for a theme that includes 2 blades, then it's just the black or dark blue frame here. And then as we look for larger and larger theme, all the way to a theme here that encapsulates essentially the entire propeller, this is shared only by very few domains, maybe 7 domains that are marked in red. And by the way, it's interesting that most of the domains here, they have 7 blades, but some of them highlighted here in bold, they have 8 blades. So on the fly, we appear to detect an evolutionary junction that links 7 and 8-bladed propellers. But notice how the same amino acid can belong to many different themes. When I talked about it with my colleague Turkan Haliloglu, this complex pattern reminded her the pattern of normal mode analysis of proteins. And this slide shows that. So this is an analysis using elastic network model of the same propeller, of the same 7-bladed propeller. And I'm showing here the 5th slowest mode and the 7th slowest mode. Now, the slowest mode, they are the functionally important modes. And you can see decomposition into parts that move in opposite directions. That in blue and in black, they are projected on the sequence of the amino acid. And you can see above them the various themes, some of the themes that were detected, and you can see that there is nice correlation between the boundaries of the themes and the boundaries between the dynamic units. You can see the same pattern also with the 7th slowest mode, and systematic analysis that we did with various examples show that this correlation between dynamics and the theme boundaries exist, and that suggests that the idea is that the themes, at least some of them, could be enchained peptides that connected to each other to form a working protein. Now, for that, of course, they need to match geometrically, but this analysis suggests that they also need to dynamically match with each other to facilitate motion in the protein that facilitates function. So I'm going back to this slide of protein space, and I'm going to argue that most of the links here between the domains are trivial. They are just between domains that are known to be similar to each other, to some degree. So we designed a way to look for evolutionary sensations, if you want, links between domains that are seemingly of different lineages. So I'm not going to introduce exactly how we did it, but this was based on an ECOD database where we looked for different groups within ECOD that are considered, it's called the X groups, that are considered evolutionarily disconnected from each other. And we asked for 2 domains, one from each X group, to nevertheless share a segment, share a theme between them, while the rest of the domains before and after the themes are dissimilar. And with that, we detected quite a few cases of we call them bridging themes, because they bridge between themes of seemingly different evolutionary links. And that includes relationship between P-loop and Rosman and other cases. I'm going to show here, I'll skip that, but I'm going to show here an example of a theme that connect 2 different proteins. You can see here one protein and another protein. You can see that the proteins are very different from each other. You can actually see that the theme is also different from each other in structure, although it is similar in sequence. And then when we superimpose, and you can see a blow up of this, the theme, even though structurally it doesn't superimpose perfectly, the theme in both cases, it facilitates iron-sulfur cluster binding. You can see it on the left and on the right. And when we superimpose it structurally, the iron-sulfur clusters, they superimpose perfectly on each other. And this is similar to what Vic showed in his talk in the joint work with Yana. And this slide is a bit technical. So we figured out that, the previous slide was based on similarity search using age search. We figured out that in a different project that Hammer sometimes give different results. And then we repeat the analysis just with the same themes, but using Hammer. And then we detected even more even more evolutionary links. So altogether, we have a collection of maybe of more than 1,000 such bridging themes. You can see their qualities. So in terms of number of residues, so on average, there's maybe 30 amino acid long bridging themes. We arbitrarily chop them at 20 amino acid to avoid random noise. You can see that the vast majorities do not superimpose well. So but while shifting between contexts, shifting between domains, very often they change their structure. And you can see that they are very identical to each other, maybe average 40% identity and very similar average, maybe 70% sequence similarity, in spite of this large structural difference. And this is an example. In the previous example that I showed, the essence of the theme was to bind iron sulfur cluster, and it was conserved. In this case, there was a theme that in one case it mediated binding to a SYM with in a Rosman, but in the other case, which is a beta propeller, you can see here in red, it lost this function. So so here the scenario was probably that this theme migrated, I guess, from the Rosman towards the beta propeller, and because the nucleotide binding was no longer needed functionally, the amino acid that mediated it and the whole segment that mediated it got discarded. You can see that it disappeared. I also have a story about adenine binding, but I think I'm running out of time, so I think I'll skip it unless questions are asked. And just to summarize, so we are studying the protein universe to understand protein evolution, and the studies suggest that evolutionary design of protein work by mix and match of those short themes, at least some of them, and we suggest that it requires both geometrical match between the themes that are put together, but also a dynamic match between them. And then certain themes mediate ligand binding, which attest to their importance. And themes can be used to predict function. If we know that certain themes are linked to iron sulfur clusters, for example, if we detect them, then we know we can predict that the protein binds adenine iron sulfur cluster. These themes are also possible for use for protein design, which Vic uses, although his thing is not called really themes, but it's a version of themes. I'd like to say that we recently published a book about origin of life, and some of what I say and much more about the early chemistry is surveyed there. And I want to thank the people who conducted the work. So I didn't get to talk about Aya's work and Ron's work, but Amit and Sergey, Caillou, Rachel, and the funding agents. I'd be happy to take questions.
Thank you, Nir. There is a question from Vic. Do you want?
I have a question. Is there anything universal about themes? Like, do they end up being compact? Do they have a low contact order? You know, is there something about them that differentiates them from other collections of elements within a protein?
No, I think themes are just a bunch of various creatures, depending on the case. So we looked at the distribution. Some of them are distributed over the surface of the protein. Some are buried. Their distribution in terms of buried, exposed, and any other properties that we could think of is not different from what you see in any protein. And actually it works, it makes sense in terms of the evolutionary pressure because they probably facilitate various functionalities. Some of them may facilitate like there would be scaffold around which the whole protein fold revolve. Some of them are important only for specific function like ion binding or whatever.
I have a question.
Yes, there's another. Oh, sorry.
So I had one question I was curious about. Maybe that's a simple answer. What we found in our work is that there is a nonlinear relationship between the sequence similarity and the alignment length that is involved in superimposing two protein structures. Do you see that same sort of signal when you're looking at the similarity that you described? Because that seems to be a very clear question in terms of relationships.
Please say that again.
Sure. So there is a nonlinear relationship between the alignment length and the identity of the alignment residues. Right. So you talked about the identity, high similarity.
I would imagine that there is, but I don't remember. I need to look at it again.
Okay, so we can talk about this then at another point in time.
Bob Hale has another question, and he was wondering what is the minimum size for a small peptide to become a theme?
Well, there's a contention here between Rachel and me. So, I mean, to be able to look back as far back as possible towards protein emergence, I would like to look at smaller and smaller segments. But then, of course, when it's too small, then we don't believe that it's significant, because I mean the level of the number of false positives probably increases. So we arbitrarily stopped at 20. We feel that below 20 it's risky, and but it's subject to negotiation. I would say, especially that. I mean, of course, this is based on very stringent threshold in terms of e-values, p-value, etc. But then the null models used to decide on these values, they are not very trustworthy, because they don't explicitly take into account a correlation between amino acids. For example, if we are talking about the helix, and there is correlation between positions on the helix. And I don't know, and I don't think anyone knows how to take them explicitly into account in constructing the model, the null model. So, and that's a part of why we are concerned, and we try to be on the safe side. We use like crazily small e-values and p-values.
So this was actually very much in line with what I was trying to ask. So I think that you may have answered it, but what I was saying is that when you have 20 amino acids to look at, you may need a lot more identity for it to be considered significant, versus if you had, sorry, go ahead.
Right, so yeah, so what we do is we build a profile for these 20 amino acids, and we search based on the profile, and that increases our confidence.
Okay. So there are no more questions in the chat. Any in the room? Paul? One sec.
Jana, good to see you.
Good to see you.
So Nir, very nice talk. Do you find within your protein archaeological search a pathway or a set of pathways that leads to an evolution of metabolism? In other words, is there some hydrogen-based metabolism that leads to a methane-based metabolism, et cetera, et cetera? I mean, certainly these proteins are not evolving in random fashion. They're evolving for an evolutionary function. So there must be some other dimension within your search pattern, which I think I didn't see.
Right, so we should look for it. There are all the major players are there, like Pilux, Roseman, flavodoxin, ferredoxin is there. We didn't do, and it makes sense to do, to look for like to date them somehow, and look for the kind of pattern that you allude to, but we didn't do it yet. The data is there, and I'd be happy to look at it together with you,
Be happy to do.
From this perspective.
I'll be I don't know when I'll come to Israel next, but it'll probably be in the summer, God help me.
Oh, yeah,
It's
good.
when it's nice and cool. All right, thank you very much.
Thank you.
All right. Nathan, we have more questions. No more questions. So Nathan-
Super, thank you.
Okay, thank you, Nir.
Thank you very much.